Welcome everyone to the second in our data dispatch series for CDAC for this spring. Today we have a very exciting talk at the intersection of climate and biodiversity and 3D deep learning. We have a team, Professor David Jablonski from the Geophysical Sciences Department and Tingran Gao from the Statistics Department, who've teamed up to do some exciting work on 3D imaging and image recognition applications of deep learning to shells. And today we're going to hear about how looking at variations in bivalve shells and shell morphology can teach us about climate and changes that are going on in climate, and also how deep learning can help us better understand these phenomena. So without further ado, please take it away, Tingran and David.
Hi, everybody. I'm David Jablonski in the Department of Geophysical Sciences. I work on large-scale patterns of evolution in time and space, integrating living and fossil data to ask questions about how biodiversity has worked in the past, is working now, and may work in the future. I'll explain what some of these big questions are and touch on where machine learning comes in. And then I'll turn it over to my collaborator, Tingran Gao, who will go into that in more detail. But first, I want to thank CDAC for hosting this event, and especially for providing the funding that made this research possible. It's a really exciting new direction for me. And I'm looking forward to continuing to do this collaboration as the work develops. So if we think about biodiversity, we can't just think about it in one dimension. We really need to think about multiple currencies or dimensions of biodiversity. Most people, when they study biodiversity, think about taxonomic diversity, the number of species, and higher groups like genera and families and so on. But there are other ways to think about it. There's functional diversity, different modes of life, and there's phenotypic diversity, morphological diversity. What we want to do is understand how these different currencies or dimensions are related to one another, because sometimes they co-vary quite tightly, sometimes they detach in time or in space, and then rejoin or continue to diverge. And if we're going to understand how biodiversity works on this planet, we need to understand the relationships among these different currencies or dimensions. And that's what our project is setting out to do. The ideal model system for this is marine bivalves, and they're really a great system for several reasons. Firstly, they mirror the large-scale trends in time and space of the entire marine biota. So they're a really good proxy for how marine biodiversity is behaving in the past and in the present day. They're taxonomically rich, about 3,400 living and fossil genera, but not too rich. That's actually a manageable number compared to, say, the essentially infinite number of insects that are on the planet and have been here before. They're insanely abundant as fossils. In fact, when I first did my dissertation, some of my study organisms were actually used to pave the roads to the outcrops I was driving to. They're that abundant, so we can really have some confidence in the large-scale patterns that we find in the fossil record. They occur at all depths and latitudes, from the high intertidal zone all the way down to deep sea trenches, and from the equator all the way up to the poles. And they have a very wide range of life habits within this single group, so that, as you may know, some oysters cement to the rocks, some scallops can actually swim by clapping their valves together and using a kind of jet propulsion by jetting water out from the valves. Some of them attach by byssal threads like the blue mussel. Some actually manage to bore into a rock, which is a very impressive accomplishment because the rock is actually harder than the shells. They can feed on many things. They can feed on detritus, these so-called deposit feeders. They can feed on phytoplankton, the so-called suspension feeders. Some of them actually have symbiotic bacteria living in their bodies. They can actually work directly from redox gradients that they live on. And some of them are even predators, which is actually very impressive for an animal that doesn't even have a head. So they do a lot of really interesting things in terms of modes of life. So what we want to do is understand the relationships among the different currencies of biodiversity in marine bivalves. To do that, we built a big georeference data set. Every one of these points represents a locality in the present-day continental shelf that has one or more bivalves recorded from it, with about a total of 68,000 occurrences. And what we can do is use each one of those points as a vertex for a convex hull that defines the geographic range of these species. And then from there, start piling up species and geographic ranges as they overlap, and then build a heat map of species richness, the number of species in every 1 degree by 1 degree cell. And that's what we see here. And there's many interesting features about this pattern. You can see, for one thing, that there's a strong gradient from many species in the tropics to very few species at the poles. And there's also a very impressive gradient within the tropics with this gigantic diversity hotspot in the tropical West Pacific, tailing off, so there's fewer species in the Indian Ocean, in the Caribbean hotspot. Really is one of the key features in modern biodiversity, has been that way for at least 20 million years. We can also look at functional diversity. Now, functional diversity is only about 42 functional groups in the bivalves as a whole right now. And you can see that they pretty much reach saturation in all of these tropical regions. There aren't that many more functional groups, just 1 or 2 in that massive biodiversity peak in the tropics in the Indo-West Pacific, and about the same number, just 1 or 2 fewer in the Caribbean. But it's still an interesting pattern, and notice that there really is a sort of a detachment between the ongoing accrual of biodiversity in the Indo-West Pacific that is not accompanied by an ongoing accrual of different modes of life in the bivalves. So we've already learned something interesting about the relationship between these different currencies. But what about morphology? That's a much more challenging currency to quantify relative to the number of species or number of functional groups. As you can see, we can do some high resolution 3D scans of marine bivalves, and we get beautiful pictures of the ribs and spines and other features on the surface of the shell. You can see there the attachment where the muscles that hold the shell closed fit into the shell, and we can really start looking at the biodiversity in terms of this other currency, in terms of morphology. That's really interesting for many reasons. For example, we can think all its ribs and spines and knobs versus the soft parts inside, and think about how the bivalve actually has to trade the investment that it makes in the soft parts that run the show versus the hard parts that protect the body. And of course, this is of more than theoretical interest, because it's the soft parts inside that humans rely on for food, and billions of people rely on marine bivalves as an important part of their diet. So as I said, it has more than a theoretical interest. And we've got a lot of data on these bivalves. These are 3D scans of about 90% of the living genera and about 1/3 of the living species. And of course, the problem is, how do we quantify that? How do we really ask quantitative questions rigorously of a data set like this? One, we can latitudinal pattern, so that we can compare the assemblage of species at the Gulf of Maine versus the assemblaging pattern here. You can see there's a wide range of morphologies in the tropics, but all the Gulf of Maine forms are basically plain to maybe a little bit of a rib pattern here, and that's about it. Whereas there are spiny, ribby, lumpy, knobby shells in the tropics. But if you think about why the tropics have so many spiny shells, the general idea is that these are defenses against ferocious predators that can break into the shells and eat the animals inside. If that were true, however, you'd expect that all of the tropical shells would be very well defended and spiny and thick-shelled, and all the high latitude shells would be plain. But that's not what we see. We see the full range of shells from very plain to very spiny in the tropics, and then a narrowing of that range as we go to higher latitudes. A very different pattern from the expectation if it's all being driven by selection for shell defensive structures in the tropical areas. So again, we've learned something really interesting by this first pass through the shells that we can find with these 3D scanners. Another thing we want to do is look at fossil material so we can understand evolution. Originally, we thought we could just X-ray these things and the matrix would separate out, but we really can't do that. The sediment is about the same density as the shell, so standard thresholding of pixel intensities didn't work. Instead, collaborator Stuart Eadie manually labeled the pixels in 6 evenly spaced slices through the CT of the shell, and then trained UNet, which is a deep learning framework to recognize the difference between the sediment and the shell material. And that software learned to distinguish those in the full array of 2,000 images to produce a digital model of the shell. You can see the shell is very flat, very elongate, and this actually represents a form of shell that no longer exists in this evolutionary lineage. Instead, this lineage lost this particular set of adaptations 65 million years ago in the mass extinction that got the dinosaurs, and never recaptured it. Instead, other lineages invaded that ecological niche, and apparently this is a really interesting evolutionary story we never could have gotten unless we were able to separate the shell from its enclosing sediment and really have a look at its morphology. So what we want to do, though, is understand how is morphology, and therefore the edible parts of the shell, going to change as climates change, because we know with warming oceans, the species are going to move, mostly poleward, but sometimes in other directions as they get trapped in cul-de-sacs, for example, like the Mediterranean Sea. This is just one of many possible maps that have been generated to show the difference in temperature at about the end of this century compared to the temperatures that were present in 1850 to 1900. As you can see, there's going to be some real warming in particular places. For example, in southern Australia, along the west coast of the US, in the northeast part of North America, which is where the scallop fisheries are. That's a very important, commercially very important. And those guys are going to really heat up and be pushed out of those waters as climates change. So that's part of how we can start thinking about the effects of climate change on how species are going to move. It's important to realize that there are many impacts on today's oceans and not just warming temperatures. Bottom trawling is present in many different regions. That's essentially the marine equivalent of forest clear cutting, really devastating to marine habitats. Invasive species are being moved around at really high rates as along shipping lanes. For example, there's one that goes through the Mediterranean and the Indian Ocean out to the Western Pacific. UV impact has really changed over time. And of course, the biggest potential impactor of all may be ocean acidification, changing pH of the oceans, which will also affect marine organisms. And the question is, how is this going to affect life in the sea? The first thing we have to do is assess intrinsic extinction risk of the species. How naturally vulnerable are they to these kinds of pressures? And we developed a simple metric for estimating extinction risk in every species in the oceans by combining their geographic range size, their temperature tolerance, which need not be the same thing as geographic range size, because many tropical species are widespread by tracking a single temperature over very wide expanses of tropical ocean. And this is unusual for most measures of extinction risk, looking at past extinction rate, asking how volatile or vulnerable to past perturbations have evolutionary lineages been, and therefore how likely are they to be vulnerable to perturbations in the near future. We can combine these into a single metric and then map them on the planet. As you can see, we just count numbers of vulnerable species. That huge diversity hotspot is the most vulnerable, with other regions, other tropical regions also having high vulnerability and cool colors, meaning lower vulnerabilities at higher latitudes. If we look at proportion of vulnerability, there's still a pretty high extinction risk in the Indo-West Pacific, especially the West Pacific. You can see now New Zealand really lights up, Southern Australia really lights up, much of West Africa, much of the western part of North America, and of course Antarctica really lights up too. There aren't a lot of species in Antarctica, but all of them are going to be at high extinction risk because all of them have been vulnerable in the past, and they're essentially, their properties make them highly vulnerable to extinction. So what we want to do is combine maps like this with our understanding of the distribution of extinction pressures, and then with a rigorous look at the morphology of these species. And with that combined, by using machine learning to quantify the morphological variety of species in these different regions, then we can really start asking questions about how biodiversity is going to be affected by climate change. What will happen to morphology, including the food value of those species? And that's over to you, Tingran.
Hi, thank you, Dave, for the nice introduction. So my name's Tingran Gao. I'm an instructor in the Department of Statistics. I'm specialized in using geometry and topological methods to study large data sets. My interest is in understanding the geometrical data in two different scales. In a large scale, we can use topological methods to understand how the data and data interact with each other. And more fascinatingly, for projects like this, I'm interested in understanding the intrinsic geometrical patterns in this 3D data. So as they mentioned, one exciting goal in this project is to give characterization of the morphological data and to connect this morphological information with a larger scale information such as geospatial statistics. Understanding how these factors such as climate change will influence the distribution of biodiversity requires capturing really many different dimensions of biodiversity, from the pattern of taxonomic diversity to ecological diversity to various different functional groups. So the diversity is intrinsically an infinite dimensional variable. This is because it expresses the form of the animal, which is just like where the rubber hits the road in some sense. It reflects a very precise interaction of the animal with its biotic and abiotic environments. Putting the numbers of quite different morphologies together, as you see on this slide, is already very difficult, which there are several major patterns of variation, which I will mention in a minute. And it is the goal of our team to set up the right tools and the right notion of mathematical and statistical background to quantify the variation in this data set. So let's begin by looking into what this data look like. After we obtain a high resolution scan of the shell, it has to be represented in some format so the computer can pass and comprehend this data. Depending on the renderer you choose or depending on the granularity you choose to visualize this data, you can transition smoothly from illustrating a surface to illustrating a surface with connectivity or just studying the point clouds. They are all stored on the hard disk using the, well, there are many existing formats you can choose from, but essentially you just have to record the XYZ spatial coordinates of every individual point, plus the connectivity information, which in the format shown on this slide is just encoding the triangle indices which are included in each triangular face. The scientific field which studies this type of polygonal data set is known as geometry processing, and the data format we just saw is often known as a mesh. The field that studies three-dimensional objects is called geometry processing. An easy way to understand what people do in this field is to put that in a parallel track alongside image processing. The modality of data is certainly very different, but you can view the goal to be pretty much aligned. There are two different other fields like computer vision and computer graphics, which we hear from very often. We can view the computer graphics as going from the direction of 3D data to generating pictures in the 2D. Well, the goal of computer vision is to interpret and understand and recognize the real 3D patterns from the 2D representations. There is a majority of development in recent years in deep learning, which is to basically pushing the boundaries of computer vision, pushing our, squeezing our knowledge to get a better understanding of what is encoded in those pixel values. Relatively speaking, the development in the 3D world is much slower, partly because the data structure is much more complex and flexible. You might have a different number of vertices, have a different number of edges, different types of connectivity across various meshes. So that's where the new developments is going to definitely require to tackle the these new challenges. But connecting this back to the statistics we are trying to do, the field of studying shapes has a very long history, at least for 100 years by now. There is more traditional approach, which is mostly landmark-based. You drop a bunch of points on each individual specimen, and you remember the number of points and remember the order of points that are dropped. And reducing the comparison of shapes into a problem of comparing just a set of landmarks. About 1 or 2 decades ago, the new field has emerged which uses more modern or more automatic methods to study these biological shapes, where the tool is mainly the more modern tool from computational geometry and machine learning. The big advantage here is get rid of the dependence on the choice of landmarks, which makes these methods much more flexible and much more scalable. More specifically, for the shell data, after the extraction of the coordinates and the connectivity, the first thing we notice is that there are 2 different types of data modalities here already. If we look at the interior part of these shells, these are the more smoother part of the shell. They are where the animal, the shell is directly in contact with the body of the animal, with the meat of the shell. These are the relatively older object of interest where it captures, for example, the volume, the weight, the size. There are more delicate version of the data which is lying on the exterior part of the shell. This is where all use these ornamentations or engravings you see. This is sometimes we call them sculpture. This is the new data set type which previous method has so far all failed to analyze, and the new methods are needed. The underlying variability comes from 2 different types, 2 different main sources. One is from the phylogeny or the evolutionary history of these animals. The other major influence comes from ecology, or how it basically depends on how the animal interacts with its environment. And the analysis goal is to understand how each of these 2 factors affect the interior shape or the exterior sculpture. The sculpture is particularly information rich because, as Dave just mentioned, there are a remarkable diversity of ecological functions associated and reflected in the sculptures. It can capture the behavior of the shell, being it a swimmer or being it a borer, or it's nesting in the ground, or it's or even more actively cemented. The shells, however, remains a relatively simple and traditional object of study. This is where the landmark-based approach can be more or less readily directly applied. There is a new requirement for how to generate those landmarks. This is where the Stewart, AD, had new developments which slices each shell into different angles and then sample generate semi-landmarks radially along those slices. Then the PCA can be performed on the connection of semi-landmarks, which we'll be able to use to be review the pattern of deformation across species or across functional groups. That's the part for the shapes. But for sculptures, that really poses a major challenge to us. The landmark-based approach is not going to work here because the real information is hidden in the periodic patterns of this sculpture. But there's no known approach that can put the landmarks in a consistent fashion which captures the varying degrees of sculpture across all the specimens. And there's a really lot of these specimens, as you have seen, connected in the data set. It's difficult to come up with an ad hoc semi-landmarking approach, one for each individual shape, and then they will immediately lose the ability to be compared. And moreover, it's a very vague question, a very chicken and egg question to begin with. What does it even mean to quantify these decorations or sculptures? What does it really mean that we have captured the variability here? So in the ideal world, there should be some major patterns of sculpture which we can relatively easily recognize. So here is an illustration of 6 major pillar types, ranging from very smooth to having very concentric patterns called comarginal, to the radial patterns which are basically rays shooting from a single fixed point. These are patterns which we can have human experts coming in and very confidently mark. But this is really the better part of the story. If only everything were as simple, it would be so much better and the task would be so much easier. Instead, let's look at what the real data typically would look like. And this is one shell. Just by spending maybe 10 or 15 seconds inspecting this shell, we can see various different patterns of a sculpture present already. If you zoom into the left part, you can see the many very small spines showing up. If you focus on the parts, these are full of the comarginal patterns. And if you inspect the right portion of this shell, you can see the radial patterns right shooting from the center of the shell to the edges of the shell. And the majority part of the shell just being smooth, there's no pattern at all. This is one example where no single majority pattern would be able to quantify, or would be able to characterize the overall ornamentation type of this shell. This is where machine learning is going enter the picture and is going to be helping us resolve this need to build end-to-end classification pipeline. Starting from 3D data, we first generate 2D topographic images by choosing a canonical projection direction and extracting pixel values which reflect the periodic oscillation patterns of the sculpture. We then send these 2D topographic images through a deep learning architecture, for example, the well-known ResNet 15 architecture, where the main difference is to remove the last classification layer, which is the in existing, for example, the ImageNet data. It is a multi-class classification problem where every image is assigned a single class, but it's not a really readily applicable for the shell data for exactly the same reason covered in the previous slide. There's no single primary type that can fit into every 3D data. There's multiple ornamentation types coexisting. So this is why we replace the multi-class classifier with a different multi-label classification head. The multi-label classification is essentially an ensemble of binary classifiers. For each individual primary type, we put in a true or false classifier, and they are not dependent on each other except for they make use of the features coming from the same architecture. After this multi-label classification training step, we remove the last multi-label classifier, and we just use the last but one output feature layer, which outputs a feature vector of dimension ranging from 512 to maybe a bit of over 1000. These features are going to be used as descriptors for the ornamentation type. So this is how we can make use of these descriptors. We assume all the descriptors has already encoded the variability we would like. We want the features to feature vectors to capture. On the left, this is a quantification of the space of shapes. This is where the PCA scores are used, and they can arrange the shapes according to the variability or the pattern of deformation across the overall shell. On the right-hand side is the feature space for the sculptures. You can see that different types of sculptures are grouped based on their proximity in the representation of the feature space. There's already a lot of information we can read off from these low-dimensional representations, and a lot of statistics can be performed using these feature vectors. A major pattern of variability is in what's known as a disparity. A disparity is essentially for each fixed class label, how much variance are there within this particular label. In these two different plots, we basically investigate the richness of the species diversity by counting how many genera are there in each family against the disparity which is in the variability in the feature descriptors. Already we can see that more diverse family tends to have a higher disparity in the feature descriptor for sculptures. But this pattern is directly in contrast with the pattern in the variability of shapes. If the family contains more genera, it's not necessarily the case that the disparity in shape will also be higher. A different type of task can be performed for telling the convergence. This is before machine learning comes into play. People tend to characterize the convergent evolution by manually or using human eye to detect the patterns of similar form or function. With these feature vectors learned from geometric deep learning, what we can do is to train a classifier to tell different patterns of shapes or a sculpture type from the phylogenetic information or from the functional groups. The convergence really happens whenever the classifier gets confused, so it's putting things in the wrong bucket, creating a more diverse confusion matrix. So basically, by counting how much score of confusion exists in these patterns, we can set a threshold and single out where the confusion, where the convergent pattern really occurs. So going forward, everything we have been doing with the deep learning architecture gives us a way to quantify the variability in a different space, which we haven't been able to address in the past. But this is just the beginning, mainly because there is a key step in the pipeline which has to choose a canonical projection direction to capture the periodic pattern. Going forward, we would like to make this process totally projection free and starting from 3D and ending in 3D. And we would like to connect this to the geospatial information with the latitude and information, with water depth information, with temperature information. And taking one step further, we would like to drop the dependence on the labels and trying to come up with architecture that will let the network detect what the features it wants to learn automatically. So these are all exciting tasks in the pipeline, which we haven't been able to do without the generous support from the CDAC. And we are definitely expecting a lot more fruitful discussion and new discoveries in science using these new tools built along the way. With this, I close this talk. Thank you very much for your attention.
Let me start with a question for David about changes in shells. I thought it was really interesting how you talked about how what you're seeing with shells kind of changes geographically as species move around. I'm wondering also, do you see changes in the shell shapes themselves? So in addition to like an existing species or shape, like moving from one part of the ocean or from the world to another, do existing shells also change shape?
Yes, they do. Although that usually is a slower process driven by natural selection. So typically when climates change or other local environmental shifts happen, lineages move around rather than evolving in place to cope with that. Let's face it, it's easier to move than it is to suffer all the different selective deaths that have to lead to a shift in the phenotypic average of a species. Of course, obviously that does happen because we see there's a lot of diversification of bivalves in the ocean today, so that must have occurred. But when all is said and done, a sudden push from the environment usually leads to an organism going locally extinct and moving around somewhere else.
Sure, that makes sense. So it sounds like the changes in the shell shapes are occurring on such a time scale that this is not something that you'd have to account for in this time scale at which you're modeling, right?
Mostly not, although there certainly are situations where there can be sort of catastrophic selection and only a certain form of bivalve actually survives. But the way things are really going to change en masse is that species populations will fade out in one place and their larvae will establish them somewhere else. For example, that's what's going to happen with the fishing grounds off the northeast coast of North America. As things get warmer, all those scallops that are being fished out that are being collected by the fishing boats out of Massachusetts and Nova Scotia are going to move up to Labrador.
Interesting. Thanks. So Tingran, I had a question as well on some of the techniques. So you mentioned you talked a little bit about dimensionality reduction and sort of clustering the different. I can't remember if it was the shapes or the sculptures, but you showed some dimensionality reduction techniques there. Yeah. And what I was wondering there is, like, often when you do that kind of dimensionality reduction, it's really important to kind of like apply domain knowledge so you kind of get the right number of clusters, right? Or you sort of aren't too aggressive in sort of removing interesting variation. And so I was wondering there, how did you, what kind of domain knowledge do you need to sort of make those kind of decisions when you're applying dimensionality reduction to that kind of data?
This is really through conversation with domain experts. So we would have to generate many different choices. For example, it's like when you are running k-means clustering, you have to choose a varying number of clusters. But then the problem is which one of these clusters makes most sense. So this is the part that we have to use the Dave's expertise as a model selector to see what kind of clusters he believes should be further apart from each other, and which cluster should be closer to each other. If that passes Dave's test, then we know our method is really capturing what people are really doing. So this is actually because we believe what's underlying whether the machine learning method will work or not is, at least in the early exploratory stage, we would like the machine to replicate what humans are able to do. And then we would generalize this pattern and scale this up and to apply some bigger data. So we try to mimic, train the network to mimic all the dimension reduction to mimic Dave's expertise at least at the total data level before we move on further as a validation.
That's super interesting. So kicking back to Dave, I mean, it seems like you're basically using Dave as the model selector, and so what constitutes a good cluster or a bad cluster in that part of the problem space, David?
So domain, for example, the domain knowledge of how big the group is supposed to be is a very useful information. For example, out of the 6 major primary sculpture types, they pointed out to us that the smooth category can be much larger than the rest of the groups. So if we see an ordination plot where the smooth part is occupying a large portion, we know it's probably a feature of the data instead of being a problematic methodological artifact. So I think this is a part where the scientific machine learning is interesting, because we are not just doing a data snooping, letting whatever the criteria that is fit best to data, but we are actually letting the nature science to play a part in creating the useful criteria.
Great.
And the
thanks.
fossil record, the fossil record actually shows us that many of the early precursors of many of the lineages we have now started as smooth, so that the different styles of ornamentation clicked in at different points in time in the phylogeny, the unfolding evolutionary tree of the group. So having that additional fossil data actually gives us new insights into the polarity of the changes and the probable frequency of the different morphologies.
That's super interesting. Yeah, I was kind of wondering because, phylogeny, those phylogenetic structures, can kind of look like a dendrogram sometimes.
That's right.
Clustering, and I was wondering if there was kind of a sensible mapping there. So it sounds like there is a little bit.
Yeah, it's really cool.
That's awesome. Thanks a lot. I'll kick it over to Rob.
Yeah, sort of following on that, I have sort of a speculative question, and I know that you haven't. You said on your next steps you'll be getting into more unsupervised learning. To train models, and kind of a question for both of you is, how likely do you think it is that this model would find features that perhaps scientists looking at these shells in the past wouldn't have discovered or used in classification?
Yeah, I can go first. So I think evaluating the likelihood of the unsupervised method to succeed is a universal question in the data science field. There's a lot of interest, but oftentimes, even before you raise this question, there is a more fundamental question, that is, when do we know we found some patterns that are actually informative? So in a sense, what we try to address with all the current work is to come up with a system to interpret whether those features capture any information. So this is actually one where we think the topographic approach should predate the real authentic 3D approach, because if we don't have a good criteria evaluating whether the features are making sense, then we can't. There's no way we can go move forward to the unsupervised setting. And I have to point out that the unsupervised setting approach it's generally treated as a black box. It would be fantastic if you just throw in a bunch of data. It just crunches data and outputs some features which automatically detected. That would be a fantastic story. But this is often almost always not the case. If you want some black box to work, you have to build this goal as a target in the black box. You have to make sure these parts already encode this pattern. So this is why we think the interpretation is going to provide us with the new guidelines to how to design those unsupervised methods. If we know what is the right way to do the interpretation, we can build unsupervised methods which will respect or highlight part of those interpretations. And then we'd have a much better idea to know what kind of a new patterns, what is a new discovery we would expect.
I do think one way in which we will make new discoveries, and I'm really optimistic about this, is in one aspect that Tingran highlighted, which are the complex sort of composite nature of the ornamentation of some of these bivalves. Biologists tend to focus on one aspect of the shell or another. Does it have spines or not? Does it have ribs or not? And I think this new way to quantify equally the different styles of ornamentation on a single shell, I'm sure will take us into something new about how these organisms are more subtly modulating the different parts of their shell in terms of different kinds of ornamentation. No one has ever quantified that in this way. And I think that's going to take us into some really interesting new avenues on the biomechanics of these organisms and how they're actually relating both to the phylogeny and to the ecology, as Tingran said.
Great. Well, thanks again, David and Tingran for joining us for today's data dispatch series. It's been really exciting to learn about this, the field. I certainly learned a lot, both about bivalves and also about 3D image classification. And it's really exciting to see these two fields come together and generate some new and interesting advances in both fields, actually. So really looking forward to learning more and seeing what comes next. The next steps look really exciting as well. Rob, I'll turn it back to you now.
Yep. Thank you both for doing the talk, and for viewers out there, come to cdac.uchicago.edu to find out about future data dispatch talks and to find links to other talks in the series. Thank you very much.
Thanks for having us.
Thank you.
Thanks.